Okay, good morning, everyone. You'll see the required disclosure slides in the screen share, and Dr. Bowman will kick off our series for today.
Okay, thank you. Welcome, everybody. My name is Gene Bowman. I'm the Clinical Trials and Brain Nutrition Lab Director here at the McCance Center for Brain Health. It's my pleasure today to introduce Dr. Jasmeer Chhatwal, who is an Associate Professor of Neurology at Harvard Medical School and a clinician scientist based at the Brigham and Women's and Mass General Hospitals. He cares for patients with neurodegenerative diseases at the Center for Brain Mind Medicine at the Brigham, with a particular focus on early onset and genetically driven Alzheimer's disease. He has a diverse scientific and educational background that spans molecular, cellular, and systems neuroscience, and his research program includes projects using neuroimaging, electrophysiology, biochemistry, molecular biology, genetics, and proteomics. His research is supported by NIH and Doris Duke charitable foundations, where he leads research on genetic, vascular, sleep-related, and inflammatory influences on cognitive decline and accumulation of Alzheimer's disease pathology. He's an investigator and project lead for the Harvard Aging Brain Study, HABS, and the Dominantly Inherited Alzheimer's Network, DIAN. He directs the Translational Neuroscience for Neurodegenerative Disease, Transcend Laboratory, part of the Ann Romney Center for Neurologic Disease at the Brigham. The Transcend Laboratory brings together neuroimaging, clinical, biochemical, and data science experts on Alzheimer's disease with the goal of developing new biomarkers that elucidate disease mechanisms, identify key drug targets for interventions, and accelerates the testing of novel therapies. Today, he's going to talk to us about how some of this work can potentially help keep us all sharp as we age. Welcome, Dr. Chhatwal.
Thanks for having me. It's good to see. Hopefully everyone is safe and warm somewhere with power. Okay. Can you guys hear me okay?
Now we
Yeah.
can.
Okay, sorry. All right. So it's quite a pleasure to be here. Thanks for joining on such a snowy day. Hopefully everyone stays safe and warm during this crazy winter. It's a pleasure to be here to talk about some of our work. So I'm going to focus on our work trying to understand why some people are at higher risk of cognitive decline as they get older, to try to understand what defines those risk states, trying to understand who's at imminent risk of decline versus people that have more baseline risk. And with the overall idea that we all want to stay sharp as we grow older, trying to define the parameters that allow us to do that and how we might approach these things, applying some of the latest technologies in biomarkers and neuroimaging to do it. So, great. So here are my disclosures. So I wanted to introduce, so this is actually, the lab is quite a bit bigger than this, but the Transcend Lab is the Translational Neuroscience for Neurodegenerative Disease Lab. So this is at the Ann Romney Center. We started this several years ago with the idea that some of the new emerging technologies, especially with respect to biomarkers and some of the new PET-based neuroimaging techniques, allow us insights into disease pathobiology, in humans in asymptomatic and symptomatic phases of disease, and really allow us to understand at the level of individual humans where people's risk states may be in terms of cognitive decline. So kind of one of the animating ideas of this lab, for me at least in terms of starting it, was to say we can now apply a lot of these new technologies to human samples, to human neuroimaging, and apply some of the latest in cognitive testing as well to try to really provide a unique insight into in vivo pathobiology, but also to understand really subtle progression during quote unquote pre-symptomatic phases of disease. So why is any of this important? Well, Alzheimer's itself has become it has long been a leading cause of death. And in terms of Alzheimer's disease as a cause of death, it's going to only increase over time because of the demographics in the US and all around the world. So our population is aging, so we have more people who are at risk. So there's around 6 million people right now who have Alzheimer's disease, and by 2050, this will rise to 14 million, and that's just in the United States. And one of the things here is that not only is Alzheimer's disease increasing in importance as a cause of death, but this is only the tip of the iceberg, because there's many people who have to change their lives in order to care for family members or other people that have Alzheimer's disease. And this represents only the tip of the iceberg for a very large public health cost and a major impact on society and public health systems. So if we're going, I'm actually going to go back to this. If we're going to be able to make a dent in this, it's going to be early in the course of disease. I think that, once we get to the point of irreversible loss of neurons and synapses, our prospects for staving off cognitive decline and for reducing this public health burden diminish substantially. So there's a lot of interest in trying to understand how we can intervene in the right people in the earliest phases of disease. And as part of this, for a long time, we've known that amyloid and tau pathology, these are the defining neuropathologies of Alzheimer's disease. Traditionally, even as much as 25 years ago, we really only saw these at autopsy. There were some hints in cerebrospinal fluid, but over time, advances in biochemical and PET-based detection of Alzheimer's disease pathology have really revolutionized the field. So if you look, these are the sort of famous quote unquote Jack curves. And what they're showing is that early in the course of disease, you have changes in amyloid-related markers. So this is CSF-A beta 42 and amyloid PET. And you first sort of see changes in these according to this formulation, and then later on tau, then some structural changes and FDG metabolism changes. And then later on in the shaded area here in green, these are the symptomatic phases of disease. So what this really drives home is that there's all kinds of pathological changes that precede the onset of symptoms. And these new technologies, including PET, allow us to have a window into this asymptomatic phase or pre-symptomatic phase of disease. And they provide an opportunity for us to intervene before there's drastic neurodegeneration, drastic loss of neurons and synapses. And increasingly, that's where the field has been moving. One thing to notice is that, when we look at graphs like that, where there's these different waves of pathological change and culminating in cognitive changes, it seems almost inevitable that if you're at stage one, you're going to progress all the way through into symptomatic phases of disease. But the reality is actually much more complicated than that. And there's many examples of this, but I'm going to show one here. This is from a paper from the Mayo Clinic, Rosebud Roberts and Cliff Jack and colleagues, putting together what I thought was a really interesting study. This is now, there's larger versions with the exact same finding that are now out here. But what this is looking at is the percentage of people who are cognitively unimpaired who develop amnestic MCI over time. And you can see here the groups are quote unquote amyloid negative. These were defined with PET, and amyloid positive. And you can see that there's a greater rate of progression from cognitively unimpaired to amnestic mild cognitive impairment in the amyloid positive group. And this is a similar figure, but looking at people who have MCI who progress to have AD dementia over time. And again, you can see that people that are amyloid positive over time definitely progress at a higher rate. But there's actually, if you flip this on its head, there's a really interesting finding underlying this, which is that many of the people who are amyloid positive actually don't decline over time, at least over this time window. Obviously, if you stretch this out further, you may see something slightly different. But the point being that many people who are amyloid positive actually are not at imminent risk of decline, at least within this time window. And even when you look at people that already have MCI, those that progress to dementia, there's quite a few that don't progress to dementia over that period of time. And so the question is, what separates these two populations? So you can be amyloid positive and at imminent risk of decline, or you could be amyloid positive and still have several years before you might show symptoms. As we develop experimental therapeutics, understanding what this risk state is, not just defining risk by amyloid positivity, but also trying to understand what other features may alter people's risk if they are amyloid positive of having cognitive decline in short order. So one of the things that's going on here is that, when you define the disease state just by amyloid positivity, you're really losing a lot of the complexity of the underlying neuropathological changes. So this is from a really nice review by Dave Notman and colleagues, where they're looking at neuropathological diagnoses that may underlie cognitive impairment across the age spectrum. And I'm not going to focus in on many things here. It's actually a very, very rich figure in terms of the sort of thoughts that it stimulates, but I'll just point out a few things. First, that there's several different pathologies that can play in, and then if you look at cerebrovascular disease, it's kind of the elephant in the room. It's in this sort of light green color over here, and cerebrovascular pathology is common across all ages, but it's almost universal as people get older in terms of the age spectrum here. And the prevalence of autopsy confirmed AD actually it reaches a peak and then it actually starts to decline in the oldest old. And what this doesn't quite, you can look at the numbers here and sort of imagine how these different pathologies may coexist. And the fact is that most cognitive decline, MCI, and probable AD cases above the age of 75 have mixed pathologies. And one of the primary things that mixes with AD pathology is vascular pathology. So this is from a neuropathological study now several years old from Julie Schneider and colleagues at Rush. And I'm not going to go through again the individual details here, but here the clinical diagnoses are shown. So these are people who passed away with no cognitive impairment, mild cognitive impairment, and probable AD. This is what was seen at autopsy in terms of the brain pathology. So this is done irrespective of what the clinical diagnosis was. These different measures were assessed. And if you look at AD plus vascular or AD plus vascular and other neurodegenerative changes like TDP-43 pathology or Lewy body pathology, the overwhelming percentage of cases actually are mixed, and vascular is the most common thing that mixes in with them. So this gives us a hint about the vascular risk may play an important role in terms of differentiating people who are amyloid positive and at imminent risk of decline from those that don't decline quite as quickly. So to try to understand this question in terms of who is at greatest risk of decline in the amyloid positive group and also in the amyloid negative group, we sought to try to use data from the Harvard Aging Brain Study. So as many of you know, the Harvard Aging Brain Study started in about 2009, 2010. It's led by two wonderful people whom many of you know, Risa Sperling and Keith Johnson. And what HABS is a longitudinal cohort study of aging and preclinical AD. The idea is that we enroll people who are cognitively unimpaired from age 60 to 90 and follow them over time with a suite of neuroimaging, cognitive testing, biochemical testing of bloods and CSF, and try to see how people do over time and what defines some of these risk states. So, there's many different kind of relevant inclusion criteria, but one thing I'll point out here is that we don't exclude in HABs on the basis of the Hitchinski score, which is a imaging measure it's a clinical measure of vascular risk. We don't exclude based on white matter hyperintensity burden or subcortical stroke, but we do exclude people who have had symptomatic strokes that are persistently symptomatic, and if people have very brittle medical conditions, they're unlikely to participate in a long-term study of this type. So that's just important to note. So this is looking at how people do cognitively over time in HAB. So this is a cross-section from a few years ago. Our follow-up is now actually closer to 10 years than 6.2 years. But what this is showing is that if you look at this line, this is saying what's the slope of cognitive change over time in the amyloid negative and positive group. So if you look in HABs, the amyloid negative group here is shown in blue. You can see that it's centered around 0. So people who are amyloid negative, this constitutes the majority of the Harvard Aging Brain Study population. Really don't change over time. There's some people that do show some decline over time over here, but still many people don't decline, and some people actually improve in their cognitive performance over time. If you overlay the amyloid positive population, so these are individuals who, according to the current guidelines, would be identified as having preclinical AD, you can see that there's many more people that decline, but actually the mode here is pretty close to 0. So there's many people, even in the amyloid positive group, who don't decline. And the simple question that we have is, what separates these people who are amyloid positive who decline from those that don't over time? And how are these folks different than people in the amyloid negative population? Are some of these other risk factors also operating in the amyloid negative group? So this has become kind of the animating thought behind the series of studies that I'm going to show here, and one of the primary things that our group looks at. And there's many different things that we could look at, and I'll talk about a few of them today to try to understand what differentiates these amyloid positive people who are declining over time from those that don't. As I mentioned, the elephant in the room and the place that we started was by looking at vascular risk factors and how they impacted cognitive decline over time and how they were related to canonical AD pathology or separate from canonical AD pathology. So now this has become sort of a long series of studies where we've characterized vascular risk using an algorithmic systemic vascular risk score, in this case, the Framingham risk score. And we simply asked, are people that are amyloid negative who have higher vascular risk factors, do they decline more over time? And if you look in the amyloid negative population, this is showing cognitive performance over time and how it changes over a period of years in the Harvard Aging Brain Study. In green are people with low levels of vascular risk, blue are average levels of vascular risk, and red are people that have high levels of vascular risk for this cohort. And you can see in the amyloid negative population, there is a differentiation, but by and large, people in all three of these groups do well. We see a different picture when we look at the amyloid positive group. And so this is looking at one standard deviation below the average cardiovascular risk, and this is looking at one standard deviation above in terms of cardiovascular risk for this population. And you can see it's really individuals who have high levels of vascular risk that do worse over time in terms of their cognitive performance. And now we have a much larger sample, and actually this effect has gotten stronger over time as more longitudinal follow-up is accrued in the Harvard Aging Brain Study. In addition to seeing this effect on cognitive decline, we can also see this effect with respect to atrophy. And as we accrued more data, we could actually also see this effect with respect to tau PET. So somewhat surprisingly, actually, we saw that individuals who had both high amyloid burden and high vascular risk showed more tau PET signal in their brains over time. And this contrasted with people that had, people had relatively high amyloid burden but low levels of vascular risk, they only had slight elevations in tau PET compared to their amyloid negative counterparts. And this was true. So this was our primary region of interest. This is looking at the inferior temporal cortex. But you could actually see the same thing in many different canonical regions where AD pathology accrues early. So these were the work of Jenny Rabin, who's now got her own lab up in Toronto, and Hyun-Sik Yang, who helps lead the Transcend lab with me. So these were some of their early studies. Over time, we accrued a lot more tau PET data, and we were able to look at this in a much more granular fashion and look at longitudinal changes in tau PET. And I won't go into the details very heavily here, but we observed that it was really, so over here, each individual is one line, and on the x-axis is time, on the y-axis is the amount of tau PET signal. So it was really people who had high levels of amyloid and either average or higher levels of vascular risk that actually showed the greatest increases in tau PET signal over time. And when we looked at this and we really drilled down on which vascular risk factors may be in play here, we found that systolic blood pressure and BMI were the primary drivers of this vascular risk synergism with amyloid in terms of tau PET. So in our formulation, at least in the Harvard Aging Brain Study, it really does appear to be the case that one of the ways in which vascular risk increases the potential for cognitive decline is by promoting tau pathology, which is a little bit different than the canonical view that there's AD pathology, that's one sort of trajectory towards cognitive decline, and then vascular risk may be a separate parallel pathway, and the two things can add together. But in our hands, it really looks like a synergism, at least in the Harvard Aging Brain Study. So what about white matter hyperintensity? So actually, the studies I just showed you, initially what we tried to do was to use neuroimaging measures of vascular risk and use those as predictors of cognitive decline. But actually what we found was that using simple algorithmic scores like the Framingham risk scores was actually a lot better in terms of predicting decline related to vascular risk. But we didn't lose track of some of these neuroimaging measures and tried to understand, okay, white matter hyperintensities weren't as strong a predictor of decline as the Framingham risk score, but clearly there's something that we very commonly see in clinic and are potentially very important aspects of brain injury injury with respect to cognitive decline. So we tried to understand what is the meaning of these white matter hyperintensities if they don't directly reflect vascular risk. And I'm not going to go into all the details here, but what we found looking, now we've looked at over 13,000 scans from different populations. Here I'm showing data from the Diane study, the ADNI study. We've also looked at the HABS study, the HABS HD study. And to make a long story short, people who have higher levels of white matter hyperintensity, often if they're along an AD trajectory, what that really means is that they're at higher risk of having CAA, and that they're at higher risk of having greater amounts of atrophy. So we found that if we looked at gray matter volume rates of change, or we looked at cerebral microbleeds, either at cross-section or longitudinally, that we saw strong associations with white matter hyperintensities. But then in many cases, in people that were amyloid positive, the white matter hyperintensities were really more reflective of whether or not somebody was on a CAA trajectory or had extensive atrophy than whether or not they had conventional vascular risk factors. So this is the work of Zara Shirzadi below, hopefully just about to get her K award. We also look at protective effects. So over here, we saw this interaction between vascular brain injury, amyloid, and tau, and neurodegeneration and cognitive impairment and dementia. But we also wanted to see whether it was possible to see putative protective effects and other lifestyle effects that may modulate some of these trajectories. So we've looked at several things now. I'll show you physical activity. So this is from several years ago, where we looked at longitudinal cognitive performance as a function of baseline physical activity level. So what this is looking at now on the y-axis is just cognitive trajectories over time in terms of how people perform on this cognitive composite. On the x-axis is time. And if you look at people who have low and high amyloid burdens, in the low physical activity group, you can see there's a big difference between people that have low levels of amyloid burden and those that have high levels of amyloid burden in terms of their longitudinal trajectories. But these two lines look much more closely aligned. There's still a difference at higher levels of physical activity, but the difference is not nearly as drastic over time. So this suggests that people that have high amyloid burden who are physically active may actually show some protection in terms of cognitive decline over time. When we looked at longitudinal gray matter volume, we saw a very constant finding, which was that individuals who had low levels of amyloid and low physical activity did show some decline in gray matter volume over time. But this appeared to be kind of the constant slow slope that we see with aging, whereas those with high amyloid burden who are not as physically active showed more brain atrophy over time. At higher physical activity, similar to what we were seeing with the cognitive performance, there was more similarity between the high and low amyloid groups if they were highly physically active. And just to note, these effects were actually independent of the cardiovascular effects I showed you a little while earlier, which was actually surprising. We thought that they would be quite collinear in terms of the physical activity effects and the vascular effects, but actually they appeared to be largely independent. More recently, now that we have much more data, this is from a paper that came out late last year, we were able to look at people who had higher and lower levels of physical activity and look at how they changed with respect to cognition, to their tau PET burden, and to their amyloid burden. And what we observed was that individuals who had low levels of physical activity, who were amyloid positive, those are in the red here, those that had low levels of physical activity declined more in terms of their cognition over time, had greater levels of tau accumulation, but that we didn't see this same effect with amyloid. So this interaction really appears to work at the level of tau and not amyloid accumulation. When we drilled down, now that we had enough data, we could really drill down on this much more than we could in the past. We observed that it was really the people that were quite sedentary. So these are the folks that have low levels of physical activity at two levels of amyloid burden, low and high. And it was the people that had low levels of physical activity who are fairly inactive that showed the greatest effect. And then this sort of graded out that at levels of moderate and high levels of activity, people that were high amyloid actually showed about the same amount of tau accumulation as those that were low amyloid. So again, this suggests that there's a protective effect here. We didn't do a clinical trial, so there's definitely more work to be done here. But both with respect to tau and cognition, this is consistent with a possible protective effect of physical activity. Another thing that we, commonly look at is, or try to understand with respect to cognitive decline, is sleep. Again, not going to go into the details here, but we have a number of studies that have been looking at this in recent years. This is a hypnogram showing the different sleep stages an individual is. This is looking at the hours of sleep. We can now measure using actigraphy or polysomnography, get a very granular measure of people's sleep and try to understand how it's related to risk for Alzheimer's disease. And this is increasingly becoming an important drug target because it represents yet another potentially modifiable risk factor for cognitive decline. So, as many on the call will know, there's different stages of sleep. So, in order to measure, these are defined electrographically. So, in order to directly measure these, the gold standard is to do polysomnography. There's different EEG patterns that are seen during different stages of sleep. Different stages of sleep are thought to be differentially related to the clearance of amyloid and tau proteins and other toxic proteins, particularly slow wave sleep. And I'll show you that in a second. So this is looking at the lymphatic system and the cerebrovasculature and asking, the activity patterns that are present in different stages of sleep may be differential in terms of promoting clearance of toxic proteins in the brain, and may be an important way in which we can eliminate some of the amyloid and tau proteins that lead to further accumulation. So several years ago, we started a study called Sonnet. So this is just finishing up now, where we did at-home polysomnogram. So here's one of our test participants with the polysomnogram setup here, with the idea that we wanted to see if sleep disruption, if we could identify aspects of sleep disruption that may be related to tau pathology, and to develop benchmarks for what we thought were sort of healthy sleep patterns that may be related to less accumulation of tau and amyloid. I'll just briefly go through some of our early findings from this study. The first was that the percentage of slow wave sleep, as other labs had seen, was related to the amount of tau pathology that was present. But interestingly, the effect was amyloid independent. So it appeared to be that the amount of slow wave sleep people had was related to tau pathology, in this case in the inferior temporal cortex, but was largely independent of amyloid burden. So this is really interesting. We don't talk about this that much, but actually amyloid burden as measured by PET or by biochemical methods only explains about 50% of the tau pathology that's present in most samples. So there's a lot more to explain, and part of that picture may be the amount of slow wave sleep. This is looking at a more recent study, so this is out for review now. This is looking at REM sleep. So one of the advantages of doing polysomnography is that we can get really good measures of REM sleep. And what we were seeing was that the amount of REM sleep, the percentage of REM sleep that people had, was related to key AD biomarkers like tau accumulation, hippocampal atrophy, and cortical thinning over time. It's also the case that we can look at day-night circadian rhythm. So this is an example of a study like this. This is led by Steph Schultz at Mass General. And what we can see is that there's changes in circadian rhythm. In this case, this is looking at relative amplitude or mean amplitude of the circadian rhythm and how it's related to age. So these are things that we know are related to age. This may also help us understand some sex differences that are present. And these can also be now, we can use these as measures that are related to tau pathology and to cognitive change over time. So that's what this is looking at here. So this is looking at the preclinical Alzheimer's cognitive composite. So at high and low levels of relative amplitude, you can see that people that had a weaker circadian rhythm, that's in the dashed line here, showed more cognitive decline over time, had lower MMSCs, more tau PET signal, and that we saw similar patterns when we looked longitudinally. So, this work is just starting now to emerge, but it's becoming part of a bigger literature that's looking at sleep as a potentially modifiable risk factor. So we're very excited to see where this goes, and Steph's doing a great job leading these studies. So to summarize, there's many different risk factors that impact the emergence of cognitive decline in older adults, including those with elevated amyloid burden. And it's not the case that everyone with elevated amyloid burden will show cognitive decline in short order. And we're now defining some of the risk states underlying this. So vascular risk, especially BMI and systolic blood pressure, appears to synergistically interact with elevated amyloid burden to promote cognitive decline, brain atrophy, and tau accumulation. greater physical activity may be protective, and we can see some of this protection at a fairly modest level of physical activity. Less slow wave sleep and changes in circadian rhythm function and REM sleep may also be tied in with cross-sectional and longitudinal AD biomarkers as well. So just briefly, I want to talk because there's so much excitement in the field, very rightly so, about blood-based biomarkers. So I was just going to show some of the work from a program, an R01 called Bionic here, where we've developed some of these biomarkers in collaboration with other labs within our system. So I'll show you a very recent study. So this is in press now, but this is now looking at longitudinal PAC. This is looking at the C2N-based measure of p-tau217, and we very clearly see people with higher levels of p-tau217 showing diminishing cognitive performance over time. It's a very strong effect. And agrees with many of the other effects that have been described in the literature. Because we have many different biomarkers available, including amyloid and tau PET, we can look also at interactions and see what the mediators are of cognitive decline. And that's a key aspect of this work, which is led by Hyun-Sik Yang, the picture in the corner. And what we find is that much of the effect of what we're seeing with p-tau217 is mediated through canonical AD pathways, meaning amyloid and tau-related pathways that then explain a lot of the changes in cognition over time. So when you look at this, there's a few things that came out of the study that I think are really, really interesting. I'm just going to focus on one of them here, which is that, so this is amyloid PET on the x-axis. This is the p-tau217 measure on the y-axis. And even in people that have very low amyloid burden, so these would all fall within the amyloid negative range, that's where that dashed line is here. Even if you just focus on those individuals and say within this group of people that has low amyloid PET signal, if we look at the different tertiles of this p-tau217 measure, we can see that it's really the people who have higher levels of p-tau217 that show us more increases in amyloid PET over time. And when we look at amyloid positivity or crossing this threshold to which what we consider amyloid positivity, people that have higher levels of p-tau217 are much more likely to become amyloid positive over time. Again, this is data from the Harvard Aging Brain Study and really drives home the point that even at very low levels of apparent amyloid PET accumulation, that you can still see gradations that are we can determine with p-tau217. So in some ways, we can say that the p-tau217 changes may actually, they seem to occur actually before some of the amyloid-related changes, although more work is needed here. And you can imagine that trying to understand this p-tau217 measure, it may be a very powerful biomarker and something that helps us stratify risk even in asymptomatic people, even in people with fairly low levels of amyloid burden. So this is clearly adding to the excitement surrounding this biomarker in the field. This is a different biomarker, again, of tau, but looking at a truncated form of tau. So many tau species are truncated. What Dennis Selko's lab, together with Dominic Walsh, found several years ago was that if you look at an N-terminal intact form of tau, it actually gives you very good signal with respect to AD and non-AD. And so we looked at the predictive value of this N-terminal tau fragment 1 with respect to cognition, neurodegeneration, and tau accumulation in the Harvard Aging Brain Study. This is together with my colleague Lei Liu. And so in the Harvard Aging Brain Study, what we observed was that people that had higher levels of this NT1 marker were more likely to show cognitive decline over time, were more likely to become clinically impaired during cognitive follow-up. And that was true whether they had high or low levels of PIB. So again, this is similar to what we were seeing with p-tau217 before, which was that there's predictive information even at low levels of amyloid, which I think is really interesting and sort of challenges the notion that all of the tau changes come after all of the amyloid changes. This is looking at now again at vascular risk, trying to define some of the brain injury. So this work was done with Steve Arnold and with Rudy's involvement as well. This is looking at VEGF-A and PLGF, and I'm not going to focus in on too many of the details. This stuff is published, but we did find that there were certain vascular markers which helped us define the brain injury state, which may be predisposing people to cognitive decline over time and to having more tau build up over time. So it appeared in this particular study that having low levels of VEGF-A was somewhat protective as compared to PLGF, which higher levels here appeared to be associated with more tau PET signal and with greater changes in CDR sum of boxes over time. This is another study again with the involvement of Rudy and several other people in his group, looking at IL-12P70 and interferon gamma. Again, what we observed was that there was an association in terms of cognitive trajectories with higher levels of PIB in individuals that had low levels of IL-12P70. And many of these findings here we're now examining in greater detail using some of the new lysoproteomics that are available now. So there's a great deal of work going on in the blood-based biomarker field. This is just a little piece that we've contributed to here, looking at higher p-tau217 levels and how they're associated with greater cognitive decline, tau accumulation, and amyloid burden, even in people that have very low levels of amyloid PET signal. And I think this is going to end up becoming a very, very crucial biomarker for understanding risk states. We talked about N-terminal tau fragment 1, which also showed some of these effects. And we're starting to define some of the vascular and inflammatory risk states from a biochemical perspective using some of these newly available tools. So just to close, I'll show you some of the more recent work from the group, which I think sort of gets this idea of how do we improve therapeutics. So the first part of this, we're really trying to identify people who are at greatest risk of decline, with the thought that these are the people that we want to intervene on with lifestyle interventions, maybe with anti-amyloid therapies. As part of this, though, we need to try to understand what makes these therapeutic strategies actually work. And this is just one example, looking at some of the new anti-amyloid antibodies here. So what we've started to look at is lecanemab. There was an interesting question that was brought up in one of our lab meetings, which is, why do we have to give so much lecanemab to people? So the dose is 10 milligrams per kilogram twice a month. That's a tremendous amount of antibody. So those of you in the lab who buy antibodies know that that's like a lifetime supply that we're giving every month to individuals. Why is this the case? We identified that there's several interactions with fibrinogen, and I'll show you this in a minute. And this also explains perhaps why we have to give so much of this drug, and also how we may modify it to make it more effective. So I'll show you that in a minute. This is looking at some of the p-tau markers. So there's many different p-tau phosphosites. People have their favorites: 175, 181, 217, and 231 all seem to share the same molecular motif. So this is from really nice work by Lei Lu and Jean-Pierre Bellier in the group here, looking at these different phosphotau sites. And again, not going to go into the details, but what we found basically was that all of the different phosphotau antibodies that looked at this molecular motif, the threonine, proline motif here, showed very similar patterns with respect to AD pathology. And I think a lot of the differences between them that people are seeing in blood may just be due to detection thresholds. So all of these antibodies could stain different tau pathologies ranging from pre-tangle to ghost tangle. And they also labeled neuritic plaques, all of them. When we looked at brain tissue using immunohistochemistry and using homogenized brain tissue for Western blots, what we observed was that for the most part, the different tau species showed you very similar patterns. This is p-tau 181 with increasing across Braak stages. But the steepest increases were really present in people that had advanced tau pathology. There wasn't that much difference at the low end between Braak stage 1 through 3. There were certain phosphoepitopes, this is looking at p-tau 231 and BA9, that showed less of a change over time, that were less consistent. But the overall picture was that these different phosphoepitopes look largely similar with respect to their relationship to Braak stage over time. And so this is now looking at homogenized brain tissue. The prior was immunohistochemistry. And across all these different phosphoepitopes, there appears to be a ramp up, so that at Braak stages 4, 5, and 6, you start to see this steep increase at higher Braak stages with these different phosphoepitope markers. We compared that also to AT8 and to a conformational antibody that just recognizes a phosphorylation at this molecular motif. This wasn't explained by the insoluble fraction, so this is looking at the urea fraction here. And so what this tells us is that, many of the markers that are based on these phosphoepitopes may really be biased towards seeing late-stage tau pathology, and just reminds us that there's a lot more to learn about tau pathology and how to recognize it in its nascent state. This is looking at lecanemab staining. So again, the study was led by Lei Liu. This is looking at Down syndrome cases in anticipation of clinical trials in Downs. But we found that lecanemab bound to both amyloid plaques and also to amyloid-laden vessels quite strongly in Down syndrome. And we've seen the same thing in sporadic AD and autosomal dominant AD, and particularly these A beta 40 rich vascular deposits really seem to have very strong lecanemab staining. So again, getting back to this idea of why we have to give such high doses of lecanemab. So this was work that was again led by Lei Liu and Jean-Pierre Bellier. We identified an interaction with fibrinogen. So it appears that at least some of the lecanemab that's given to people is likely bound to fibrinogen, and this affects how much may be available in the free state to move across the blood-brain barrier, and may help explain why we have to give such high doses of lecanemab. So this work is still in its early stages, but it's been very, very interesting to look at, and brings home an important point, which is that the free drug, the amount of free lecanemab that's available, really is the defining feature for how much is available to go into the CNS compartment, and that the bound drug itself may not be able to exert the desired pharmacological effects because those occur in the CNS. And so we estimate that around 90% or a bit more of lecanemab may be bound to fibrinogen. This is important for a number of different reasons. So without going into the detail about how we identified fibrinogen as a binding partner, fibrinogen is actually present in AD brain, and you can see a lot of astrocytes that are laden with fibrinogen when you look at AD brain tissue. Fibrinogen is also a key part. Deposition of fibrin within blood vessel walls is also a key part of the development of CAA. So we think that this interaction may be also very relevant to ARIA and to CAA progression. And we can actually try to understand more about this interaction by modeling the antibody, understanding where lecanemab may be binding to amyloid, and then trying to make selective mutations to see if we can create versions of lecanemab that are more or less likely to bind fibrinogen. And so this is just a little Susan of some of this work. So this is looking at brain tissue. We made several different generations of mutations within the lecanemab protein. And without going into some of the detail, you can actually alter by making particular mutations the relative binding to fibrinogen versus amyloid plaques versus amyloid-laden vessels. And so we think that there may be some tools here to help us understand what the fibrinogen interaction with amyloid and with lecanemab, how that might be relevant to some of the therapeutic potential of these antibodies, and hopefully to develop safer versions of these antibodies. So just to summarize this portion, so lecanemab dosing may be impacted by interactions between lecanemab and fibrinogen. We observed that lecanemab binds to both parenchymal and vascular amyloid in deposits in sporadic and Down syndrome. Mutations in the antibody sequence itself can change the binding to parenchymal versus vascular amyloid deposits, as well as the affinity to fibrinogen. So a lot more to come on this, but really interesting kind of initial results. With that, I wanted to leave a lot of time for questions, so hopefully I've done that, but I really wanted to thank, it's a fantastic group of people at Mass General, at Brigham, and many thanks to my lab, and especially thank you to Dennis and Risa for being fantastic mentors and really supporting a lot of this work. So with that, I'll take any questions people might have.
Thank you very much, Dr. Chhatwal. That was a remarkable presentation and body of work, actually, from the entire group on your end, looking at vascular factors and a whole host of different important questions to help us understand the natural history of these diseases and different pathways that contribute, and the development of new biomarkers that will help us identify and detect the disease process earlier and intervene earlier. With that, we'll open up with a question. Dr. Kanzi.
Jazz, that was an amazing talk. Really appreciate, obviously, the time and effort you put in putting this together. And you made an awful blizzard snow day a great day with that talk. So Thank you. I got a couple of questions. First, on the last part, you know, the people forget that the APP with the splice domain for the Kunitz protease inhibitor that we described in Nature in 1988 is actually protease nexin 2, which is part of the clotting pathway together with fibrinogen. And that's really probably the best role we know for APP, other than being a precursor for A beta. The secreted Kunitz APP works in coagulation.
Mhm.
And beyond that, you probably already know that A beta binds fibrinogen directly. There's a work of strip matter at Rockefeller. I think the KD is in the like 20 nanomolar range for A beta to fibrinogen.
Yeah.
And I'm just wondering, is it possible that you're getting an ARIA effect when you have lecanemab or a monoclonal antibody binding to A beta that's already bound to fibrinogen, creating then some level of inflammation and vasogenic edema by the combination of the binding to the two versus just, say, CAA alone?
Absolutely. And we've been very struck by that interaction as well. So we've looked at you're right. There's a nanomolar range affinity for A beta species with fibrinogen. So we're going through and looking at different A beta monomers and dimers and seeing what their affinity is now with SPR. So that's work actually, hopefully we'll have some results on relatively soon. I do think it's something that when you're talking about nanomolar affinity, that's incredibly strong interaction between A beta and fibrinogen. So there's actually two questions that are embedded there, which is that what's the physiological relevance of the fibrinogen A beta interaction? And then in a therapeutic context, how does that explain some of the interactions with lecanemab with fibrinogen and lecanemab with fibrinogen bound to A beta? So those are difficult questions to sort of disentangle, but really important ones. To me, it's really striking that we're seeing these fibrinogen patches. I don't have it up on the screen right here, but maybe I can go back. But to me, when we, first saw some of this immunohistochemistry, I mean, if you really look at some of these vessels, it really might be the case that fibrinogen is maybe binding A beta there, and maybe part of a repair pathway at the sort of brain vascular interface that we can try to understand better. There's a lot more to do on that front. I think that, as you point out with your question, we don't know all of the functions, the physiological functions. In fact, we have a lot to learn about some of the normal physiological functions of APP and A beta peptides. But I do think that this interaction ends up becoming, I think, a very important one to study, and we're putting a lot of energy into trying to do that. So I don't have a ton of answers for you right now, but I do think that it could well be the case that the fibrinogen here that we're seeing is actually there to bind up some of the A beta, which may be infiltrating the blood vessel walls. So again, any thoughts on that are more open to. Yeah, go ahead.
I mean, two things. I mean, it's an awful coincidence that secreted APP with the KPI domain is protease nexin 2, which is the main coagulation factor that hits factor 9A, 10A, 11. And then you have A beta binding fibrinogen. Apparently, when the A beta fibrin complex creates a much stronger, coagulant, so to speak, that it's not as prone to dissolution by plasmin. So A beta helps create. So I'm just wondering, you just got me thinking, could it be that when you see vasogenic edema with lecanemab, that you have some type of very strong complex between A beta and fibrinogen that plasmin can't break down, and then the antibodies binding to that and then creating disruption along the vessel wall? I think you're onto something pretty potentially amazing there. And I have one other question. I don't mean to hard, but I'll just get out of the way. So p-tau 217, going back to the work in our unit with Du-Young Kim and his group, showing that it's A beta oligomers in the 3D neurocell culture models. It's really the A beta oligomers driven by the 42/40 ratio that drive p-tau and tangle formation in the neuroculture model, the 3D neuroculture model, more than, let's say, fibrils and plaques. So I'm just wondering, is it possible that what you're seeing with p-tau 217, when PET A beta amyloid is down, is you're seeing that oligomers accumulating it can't be, you don't have a lot of visible amyloid burden and plaque yet by PET, but there's a oligomer that's accumulating earlier on and driving that p-tau 217 prior to seeing amyloid by PET scan. I'm just wondering your thoughts on that.
I mean, absolutely. I think that, in a way, as you know, lecanemab was actually developed against quote unquote protofibrils against the antigen from the Arctic mutation. And I think that it does drive home the point that amyloid pathology, we sort of orient to the visible, what we can see with PET, but there's a lot going on besides that, and the toxic oligomers that are present with A beta can be driving pathology themselves, even if visibly we don't necessarily see that difference. So, and I actually think that the data from Diane really supports that, which is that there's huge variations in apparent amyloid burden. If you look at amyloid PET, even in people that have, you can see low levels of amyloid PET signal in people with autosomal dominant AD. So the Arctic mutations is a extreme example of that. But I think very much it's the case that we're missing the actual toxic species and some of the interplay, which may be driving especially early AD pathology that's there. So I absolutely think that's true. And so we can look at that, and we will hopefully in Diane and then soon in sporadic. Yeah.
Yeah. Great. Thanks again. I'll shut up now, Gene.
No, it's okay. It's a great discussion. Okay, Dr. Dodge.
Hi, Jasmeer. Such a wonderful presentation as
Thank you.
always. So touch so many wide variety of topics and then very deep. One thing I thought is very interesting is that physical activities. I always thought physical activities, cognition's relation is sort of mediated by hypertension or white matter hyperintensity is a big one to be mediated, but you found these are independent. It's not mediated by white matter hyperintensity and other stuff, but mediated by more like tau. So what do you think about this underlying mechanism, physical activity versus this tau mediation? What's the biology
Yeah.
here?
So there's a few pieces there. First off, I have to admit that I thought it was also going to be collinear with hypertension, with BMI and other things. And there is a relationship there, but it doesn't explain the association with cognitive decline and with tau. I think that the short answer is that it is your physical activity. I think the way that we're measuring it, we're not intervening on physical activity, and we're not changing people's physical activity. We're simply observing it. So first, we need better trials to try to get at this, to make sure that what we're seeing is not reverse causation with respect to physical activity. I think that's an important feature. We also need to look at the interplay with sleep. So it's possible that it alters circadian rhythm or alters sleep patterns in a way that favors clearance during sleep. I think that that's possible. Neuroprotective mechanisms in terms of brain volumes. We haven't seen enormous effects with respect to brain volume, but we have seen some, with meaning that there's the people with higher levels of physical activity for a given amount of amyloid will have better brain volumes over time. But those are not huge effects. They're not as big as they were with the vascular risk factors. But I think that there's two possibilities. One, that there's neuroprotective mechanisms in terms of promoting survival of neurons and synapses, and that there's enhanced clearance. I think those are kind of the two things that sort of pop out to me. We do need to make sure about reverse causation. The last piece is inflammation, which is a hard thing to define. A lot of the physical activity markers that we've looked at here do appear to be related to inflammatory markers, even outside of associations with BMI. So that's another place that we're sort of kind of building out this story. But the real answer is we don't know. What it does suggest, though, is if we're going to do clinical trials using physical activity, focusing on people that are sedentary and amyloid positive may give you the most bang for your buck in terms of seeing an effect. So on the clinical side, that's what I'd like to see, what happens. And on the sort of mechanistic side, understanding these interplays with sleep, circadian rhythm, and inflammation is, I think, important.
Thank you. Maybe physical activity, tau relation, in the trial, somehow we can measure this underlying biology. That would be fascinating.
Okay, thank you for the question, Dr. Dodge. Dr. Mehdi, I believe, has a question.
Yeah, great talk. Thank you. Another question regarding the interaction of the lecanemab and the fibrinogen. The first question, do we know how much of the lecanemab get across the blood-brain barrier? The reason I'm asking this, does the interactions happen before the lecanemab enter the blood-brain barrier, enter the brain, and how much of that interaction happens after the entering the parenchyma. The second question, again, regarding this fibrinogen, does aging have a role in this? Does the fibrinogen in aged people is kind of promoting these interaction between the lecanemab and fibrinogen, something that has, like, I don't know, with the chemistry, or how sticky is it, or something changed? Has anyone looked into how the lecanemab interact with the fibrinogen from healthy people? Thank you.
Yeah, no, those are good questions. As is the case with most antibodies, the percentage of the infused antibody that crosses the blood-brain barrier is very low. Even with the brain shuttle, you're talking well less than 5% of what you give crosses the blood-brain barrier. Actually, it's significantly less than that. It's less than 2% actually crosses. And so I think that it is the case because fibrinogen is in such high quantity in the blood. I think it's the seventh most abundant protein in blood, that there's a lot of potential for interaction. And even though the affinity of lecanemab for fibrinogen is relatively modest, the amount of fibrinogen that's present is really important. So, I mean, I think the way to look at that is to try to understand how effective lecanemab is in clearing in amyloid people as a function of their fibrinogen levels. That might be one way to do that, to try to get an idea of, do we have to vary the dosing based on the amount of fibrinogen that's there? The approach that we're trying to use, it's tricky, but we would love to be able to come up with assays that separate the free versus the bound fraction of lecanemab. And so, we are working towards that. Once the lecanemab crosses, that's a different question in terms of the interaction. I think that the amount of A beta that's in the CNS is high. So you get a flipped peripherally, there's a lot more fibrinogen in the blood than there is A beta. Centrally, that flips. And so I think that those are kind of separate questions in terms of how that might play out. But long term, how lecanemab's interaction with fibrinogen affects vessel remodeling, I think, is really important in trying to understand how it may be promoting CAAs there. So another thing we can do is use some of the mutants that I mentioned of lecanemab that have greater affinity for fibrinogen versus A beta, and use those in animals and try to understand which of these are actually effective in clearing amyloid burden and which ones aren't. So we can use a functional outcome, in this case, looking at amyloid burden cleared by the antibody, to try to understand this. So we're kind of doing all those things in different buckets. On the clinical side, understanding if fibrinogen levels are relevant to dosing. On the biochemical level, trying to characterize the strength of some of these interactions on different compartments, and then using some of our mutants for some functional assays.
Okay, thank you for the question. I have one, Jazz, for you. It's related to p-tau217. We're utilizing this in our early phase trials currently, as a lot of people, I believe, are now at this point, to enrich study populations with some pseudo-AD pathology. But at what threshold for the blood-based p-tau217 do you think is significant? I see some of the studies you've seen that people have significant changes over time at a certain threshold, but do you have a range for p-tau217 in the blood that you think is a real critical time point? I know it's a little bit of a moving target depending on who the population we're looking at.
You know what the thing is with, so it's a really good question, and one that's going to be important these are already really important biomarkers in clinical research, and they're becoming increasingly clinically important in terms of diagnosis and monitoring therapy. One frustration I have is that these different assays, when they're run on different platforms and using different antibodies, can have very different thresholds, right? You can imagine that certain assays are going to be more sensitive, other ones are going to be less sensitive. The sort of linear ranges for them are also going to be very different, and how they operate in different population. So there may be certain assays that are really good in preclinical AD and thresholds that are important to use in preclinical AD that might be very different from what you're using in symptomatic AD. So right now it's the Wild West. We're kind of doing all of the above. There's different antibodies. And the other thing is that many of the antibodies are tied to specific platforms. So, if you really want to run, the Johnson & Johnson assay, the Lilly assay, and other things, you can't always access those antibodies. So actually what we're doing, so Lei Lu has developed a series of very good calibrators that will allow us to compare across the same sort of range of p-tau across many different assays and trying to come up with some standardized units. So we have an R1 that's in for review now that really tries to do that, to try to come up with assay independent units for how much tau is there, and then try to compare first how the thresholds are similar or dissimilar across the different assays, because there may be some that are better in preclinical disease, for instance, versus symptomatic and other things, to try to understand that. So we're still trying to figure it out. Right now, most of the cutoffs are assay specific or are there to maximize AUCs, separating AD versus non-AD, but that's actually a fairly superficial question in some ways and very dependent upon the population that went into the derivation of that threshold. So to try to get around that, coming up with standardized units, I think is the first step in answering the question that you asked. So we are not even at that step yet.
Okay. And before I get on to that, I hope everybody has a few more minutes here because it's a snow day. I just wanted to mention that we have developed and validated a panel of 150 nutritional biomarkers. I know you looked at physical activity in the HABs and the Harvard Aging Brain Study. If there is interest in looking at an objective indicator, dietary intake and metabolism related to these outcomes, we have the methods to do that. We're have been contacted by the U.S. Pointer Trials PI, and we're putting in an R1 in June to tap into U.S. Pointer for those. But if there's interested in the Harvard Asian Brain Study, we'd love to collaborate. And I'll just point your attention next to Dr. Duquesne's question in the chat. Is Du still with us? He may not be able to be online. I can read his question there. Are you able to see that, Jazz?
Yeah, I've got it. I wonder whether modulation of interaction between lecanemab and fibrinogen affects microglial A-beta clearance. Did your reduced fibrinogen binding lecanemab have similar A-beta clearance capacity in animal models? So that's a good question. I think that we just have to test it. We don't know yet. We can when we do the immunohistochemical assays, we can see that there's a preference more for plaques than for vessels when we look at that particular mutant, but we don't know how effective it's going to be in terms of clearing amyloid, and that's clearly a place to go. That's where we're going with it pretty quickly. And that's, go ahead.
Actually, I have a question following up exactly. So what is the site for the fibrogen interaction with antibody? Because FC region and these are definitely physically close to each other, and then we can expect some interaction. But if they are far away, probably it's different. Where is the interaction site of antibody with fibrogen? Are they close to variable regions or FC regions?
Yeah, it requires the variable region. So it's not an interaction with the FC, so it has to do with the variable region of lecanemab itself. The mutants that sort of showed, so when we actually go directly into the variable regions, Jean-Pierre has made a series of mutations that made a series of mutations within the variable region. Those tend to abolish all binding across the board. But when you make more subtle mutations in the sort of accessibility of that variable pocket by making mutations adjacent to but not in the variable region, you can start to see some of this tuning more towards fibrinogen or more towards amyloid, which is really interesting and exciting. Yeah.
Very interesting.
A beta binds the C-terminal region of fibrinogen. So I don't know if that helps. I think it binds like 600 or so onward out to the C-terminus. So I'm not sure how that affects things.
Yeah. We've actually been trying, based on the sort of some of the questions that you're asking, it sort of seems like you're interested in some of the same things, which is that it might actually be possible to make lengths of A beta that are not normally seen physiologically, but may be very efficient for displacing A beta from fibrinogen. And if we do that, we might actually be able to do several things. One, it may improve A beta biomarkers in blood, because at least some of the A beta that's present in blood is probably bound to fibrinogen fairly tightly, right? That it's a very strong interaction. And so what we're trying to see is there some way that we can play around with the fibrinogen interaction as a way to improve biomarkers? It may also be something that could be affecting therapeutics, because you could imagine having a blocking peptide and other things. So we're definitely trying to work on that. And any ideas on that, or any people who want to work on that, we're very interested in it. There's a lot there actually to study, and it's very relevant to therapy. So.
Okay, thank you for the discussion. I think we have one more question from Dr. Zhang in the chat regarding aging-related A beta pathology changes that you showed in Habsburg. Can you comment if they are due to A beta generation and/or clearance?
Yeah, I think that yes, first off, it's great to hear from you, Martin, and thanks for working with us on some of the inflammatory projects. So it's great to hear from you. I don't know. As you know, the literature suggests that it may be due to deficient clearance more than it is to changes in production. But I think that data is actually fairly thin. Most of it, the most compelling data of that type seems to come from silk studies. So this is the stable isotope labeling studies that Randy Bateman and others have been doing for a number of years. But I think that clearance is a reasonable thought. That's one of the reasons why we got into the sleep literature. So there's a lot of age-related changes in sleep. So you can definitely talk yourself into a mechanism where there's an age-related change in sleep, which leads to reduced clearance, which leads to greater accumulation. You could definitely talk yourself into it, but it's a hard thing to sort of separate out each one of those individual pieces. And practically, this idea of clearance is it's a seductive idea. It makes sense too that there is a mechanism of clearance. What that actually looks like, are we clearing things by breaking up the molecules? Are we doing it by moving them to a different compartment, say from CSF to blood? What is the nature of that clearance is actually a very complicated question. So I don't have all the answers to it, but you could definitely talk yourself into a clearance story based on the silk data and interactions with sleep for sure.
Okay. Well, again, thank you so much for joining us today, Jazz. It's really remarkable presentation, very intriguing, lots of important work done and completed, and we hope to have continued future research together. So, and also welcome as a new affiliate to the center, to McCance Center for Brain Health. Thank you all, and stay safe out there and have a great week.
Yeah, stay safe and warm, everyone. Thank you.
Okay. All right.